I wish to compare the basic strength of this 0.1 mole per litre sodium hydroxide solution and this 0.1 mole per litre sodium carbonate solution. To do that, I'm going to make a quick batch of red cabbage solution, which is actually a natural indicator. To do that, I simply need 1 or 2 leaves of red cabbage. This is red cabbage available from your local grocery store. I just need to tear up these leaves into small pieces. In front of me I have a beaker of boiling water. Just break it into small pieces. The smaller the pieces, the faster you get the pigment out of the red cabbage. You place those into your hot water and stir. You'll notice the water instantly turns blue. I've now allowed this red cabbage solution to sit for an hour. In that process, the dye from the red cabbage has actually gone into the water, dissolved into the water, and the solution is cooled. So now it is cool enough for me to hold and to pour into a plastic or some other container for storage. When you transfer it into the container, just make sure you don't get any of the leaves in there. Note the colour of the solution. This is the colour of the red cabbage indicator at neutral pH. And label that. Now I've taken the liberty of actually making some more red cabbage solution, and just to prove that it's safe, cheers. Ugh. Not very nice. To test the properties of this red cabbage natural indicator, we're going to use some household substances and look at the colour changes of the indicator that occur. In this first test tube, I have a 1 mole per litre solution of sodium hydroxide. Sodium hydroxide is a household substance. It's actually used in Drano. It's also used in cleaning grease off wheels. Okay. I add a generous squirt of my red cabbage indicator solution to the sodium hydroxide. Takes a while for it to equilibrate. Sit that down. You form this nice bright yellow colour. Okay, it's quite impressive actually. Okay, next I have a solution of 0.1 mole per litre sodium carbonate solution. Sodium carbonate is also known as washing soda. Add a generous squirt. Allow that to equilibrate and sit that down. And we note that in a strong base, sodium hydroxide, the indicator turns yellow. In the weaker base, or it's a moderate strength base, we get this nice green colour. In the next test tube, I have some solid baking soda, sodium hydrogen carbonate. This is a solid, so I add more indicator. I don't need to shake that. It's quite apparent that you get this nice blue colour. We go from weak base, stronger base, and even stronger base. We go from blue, green, to yellow. In the next test tube, I have tap water. Tap water is neutral, and in fact I get the purple colour of the neutral red cabbage indicator as it's produced. In my next test tube, I have vinegar. Vinegar is a 5% solution of ethanoic acid, a weak acid. I add my indicator, and I get a pink or pale red colour. In my final test tube here, I have a 1 mole per litre solution of hydrochloric acid, a strong acid. We can see that there is a difference in those two colours. This one is more red relative to the pink of the ethanoic acid. We take that as a series. We note that in strong acid, our indicator is red. There's a transition to a blue form of the indicator in weak base. Purple and pink are obviously a mixture of the red and blue form of that indicator. And lastly, there is a transition from blue to yellow. As we go from weak base to strong base, we get a mixture of yellow and blue, which results in green. So there are 3 forms of this indicator: a red form, a blue form, and a yellow form.